Am I audible? Yes, very good evening, Satyam. Thank you. I'm very well, doing very well. I hope you all are also doing good, and your preparations are on full swing, because we just have a week or two now left for the final exams. Okay. Now today's topic is from Unit 1. Let's look at the rest of the portions. Mostly it will be the metabolism, and apart from that, some concepts of protein, amino acid, a little bit of bonding also. Okay, yeah, very good evening, Tulika. Okay, first time on YouTube. All right, so we welcome you to our channel, Biotecnika channel, and I can guarantee you that this is going to be very beneficial. Now that you have found us on YouTube, do subscribe it and follow it, and regularly you will be getting a lot of updates regarding the current events, notifications, important exam updates, and even the educational videos. Okay, yeah, you can mail at info@biotechnica.org, Tulika, if you want to speak to any of the faculty members of Biotechnica, if you want some academic guidance. Yeah, so in order to speak to the faculty members of Biotechnica, I'll have to mail at info@biotechnica.org. Okay, then it will be passed on to the faculty members, and probably they can schedule career counseling session also for you. But for that, first you have to contact our student support team. Our toll free number and email ID are all available on our official website, Biotechnica. So kindly contact them, and they will fix up a career counseling session for you. Okay, so the first concept for today will be from metabolism, glycolysis, which is carbohydrate metabolism. This question is from December 2023. In a mammalian cell undergoing active glycolysis, what would be the effect of a sudden increase in the different concentrations, that is AMP, citrate, ATP, and glucose 6 phosphate? Okay, so which one of the following representation represents the combination of all the correct statements? So whether the increased ATP will inhibit glycolysis or increased AMP will stimulate glycolysis, what is it? Tell me. Okay, Satyam says option 2. Let's see what others have to say. Yes, all those who are attending, I want answers from everyone. Just try it. Okay, Sarita says option 3. See, remember why we have glycolysis. What is the ultimate result of glycolysis? Think about that. Yeah, sure, Tulika. That's a very good decision which you have taken to prepare CSIR NET. And just stay for some time. I'll be giving you the details of a batch which is now, it will be for June 2025 exam. Okay, it's the Lakshya batch. You can join that batch and you can prepare that for that exam, and even it will be beneficial for you for the next exam that may be in September or maybe in December. Okay, now the increased ATP, see, the idea of glycolysis is to produce energy. The whole idea of catabolic processes, glycolysis is a glucose breaking down into pyruvate, right? And for that, this is a catabolic process. Catabolic process means it will break down bigger molecules into smaller molecules to release the energy. Okay, now, increased ATP will inhibit glycolysis. Think about it. Ultimately, from glycolysis, when pyruvate further enters into the TCA cycle, when it enters the oxidative phosphorylation, ultimately ATP will be generated. So, if already ATP is present, then what is the necessity of glycolysis? In that case, glycolysis will not take place, isn't it? So this statement seems correct. Increased ATP will inhibit glycolysis. Yeah, you're most welcome, Tulika. Increased AMP stimulates glycolysis. Now, ATP and AMP, they are actually in the same pool. Okay, we know AMP converts into ATP, ATP converts into AMP. If AMP is high, it means ATP is less. Okay, so if ATP is less, then it is an indication that energy is not there in the cell. Okay, or you can also think about it, AMP is high means it's an indication that energy is required in the cell. In that case, bigger molecules should be broken down into smaller molecules to generate energy for the cells to function properly. So that's why increased AMP stimulates glycolysis. This is also a correct statement. Okay. So that's it. You have got A and B. Third is the right option. Yes, very good, Sarita. Your answer is right. Increase citrate. Now, how do you get citrate? This pyruvate, as I said, it enters the TCA cycle. Pyruvate, it combines, first of all, it converts into acetyl CoA. Okay. And acetyl CoA will be converted into citrate in the TCA cycle. So, citrate is the first intermediate in TCA cycle. So, this TCA citrate, if it is present, that is also an indication that TCA cycle will proceed. And if TCA cycle is proceeding, it means that energy will ultimately be generated. So then also we don't need glycolysis. Glucose 6-phosphate. Glucose 6-phosphate is one of the first products. It is the first product of glycolysis. The glucose is converted into glucose 6-phosphate, and glucose 6-phosphate has a feedback mechanism on its own enzyme. This enzyme which produces this, it is hexokinase, and in liver cells it is glucokinase. Glucose 6-phosphate will have a feedback inhibitory effect on hexokinase. If too much of glucose 6-phosphate is there, then the glycolysis gets inhibited at the first step itself. So this is also wrong, which is stimulate glycolysis. No, it will be inhibit glycolysis. Okay. Increased AMP, already we saw it stimulates glycolysis, so inhibits glycolysis is wrong. So that is why A and B is your right answer. Yeah, welcome Rinku. Now, the next question is from December 2017. This is a match the following question, but this is a slight tricky one. You don't have all four, means 4 and 4. Okay, one of that will not be the answer of these options. One of them will not be the answer. So such kind of match the followings are little tricky, and if you have a third column also, then they are even more trickier. So it will be like match one of this, one of this, and one of this. So that will be even more trickier. Okay, so don't think that if it is match the following, definitely it will be easy and you'll be able to solve it. All right, so can anyone give me the correct answer for this? This is in case of oxidative phosphorylation, which involves the electron transport chain and the ATP synthesis through the ATP synthase or F0F1 particle. So here there are some components which will have some inhibitory effect on the components of electron transport chain or maybe the ATP synthase. Okay, Sarita says 2. What about others? Do you have any other options? Or think about it. What can be the correct option here? No one? Okay, see this inhibitor and activators overall in CSIR, they are very important. All right. Whenever you have to, whenever you have come across any such, even in unit 2, let's say, this microtubules and microfilaments, what will activate their polymerization or what will inhibit their polymerization? Even those kind of activators, inhibitors in plants, also the different herbicides that we use, this kind of questions, this kind of information you should always remember because questions often come from here. Okay. Now, if you know any one of this here, particularly this one is easy, that oligomycin, it inhibits ATP synthesis. Okay. ATP synthase, especially it will bind to the F0F1 particle. Okay. Because all the others, if you can see, this is the adenine nucleotide translocase. In the inner mitochondrial membrane, ADP goes in and ATP comes out. So those nucleotide translocases can be inhibited, or the cytochrome BC1 complex. It is one of the complex of electron transport chain, or FES cluster to ubiquinone, which is a component of complex 1. Okay, disrupts inner mitochondrial membrane potential. So this is again some kind of, you can say, um, this dinitrophenol type of molecules, which is an uncoupler. Okay, so any of this doesn't match with oligomycin, so B will be 1, and you don't have any other option in which B is 1, so that's why option 2 is the right answer. Okay, therefore the correct answer is 2. Yes, yes, that's right, Satyam. Now, talking about rotenone, rotenone is a very important kind of molecule which is popularly known, most common information, that it inhibits the complex 1. Okay, ubiquinone is the mobile carrier, whereas complex 1 is the first complex. So complex 1 has this iron sulfur centers, FES centers, which pass the electrons to ubiquinone, and this process is blocked by rotenone. Okay, then we have the antimycin. Antimycin will be blocking the BC1 complex. It is again a common inhibitor of BC1 complex. And valinomycin, it will be inhibiting the inner mitochondrial membrane. Okay, sorry, it will be disrupting the inner mitochondrial membrane potential. Okay, valinomycin. Because, so you will be seeing over here the different kind of inhibitors. It's advisable that you learn all of them. ATP synthase, you have oligomycin plus DCCD, and when the protons are passed on to the intermembrane space from the different complexes, that is complex 1, 3, and 4, the BC1 complex, which I was talking, it is the complex 3, and cytochrome C oxidase is the complex 4. Okay, complex 2 is the succinate dehydrogenase, which is also an enzyme of the TCA cycle. So this third complex, or BC1 complex, is blocked by antimycin. Complex 1, along with rotenone, it is also blocked by some other molecules, amityl, demerol. Just remember these names. And complex 4 also has very common type of inhibitors. Most of them you would have already heard: cyanide, carbon monoxide, and azide. These are popular ones of complex 4 blockers. Yes. Yes, that's right. That's right, Satyam. Yeah. Oh, you are typing so much. Okay. No need to type, Satyam. You can just tell the option number. Okay. Otherwise, it will be painful. No, so type so much. It's okay. Now, the uncouplers, see here something is given as uncouplers: decumarol, FCCP, or dinitrophenol, which is also called as 2, which is also called as DNP. Now, what they do, see, when the protons are transferred to the other side of the membrane, this is the intermembrane space, and this side is the matrix. So, in the intermembrane space, you will have an abundance of H plus ions. Okay, now this H plus ions, they will be giving, or they will be generating something called as a proton motive force. All right, proton motive force means these protons will try to come back to the other side of the membrane, that is back to the matrix from where it primarily went outside. But these protons, they cannot cross the membrane. They have to come through the F0 F1 particle. This is the ATP synthase. This is the F0, this is F1. So they have to pass through this one only, because of which conformational change will be there and ATP will be synthesized. Okay. But what does this uncoupler do? Even if the protons are generated, okay, the uncouplers will sequester these protons on the other side. Sequester means it will bind them and it will hide them. Now, if the H plus ions are not present, then what will go through the F0 F1 particle? Isn't it? So, if ATP synthesis in that case will not take place. Okay, but electron transport chain will keep on taking place, thinking that the gradient is not building up. Actually, H plus ions are transported, but because of the DNP use, there is the gradient of H plus ion is not able to form. And if there is no gradient, then there is no proton that can pass through the ATP synthase. That's why there will be no ATP synthesis. So that's why DNP is called as uncoupler. All these are called as uncoupler, because generally the electron transport chain, the proton chain, proton movement They are all coupled with the ATP synthesis process. If ETC happens, then only the H plus ions will be passed. Then only ATP synthesis will happen. But in this case, the two processes are not connected anymore, because even though the electron chain is continuously taking place, proton gradient is not able to be built up, and that's why ATP synthesis is not happening. Okay. Based on this, you have this June 2016 question, which is a very interesting question. Can anyone try to solve this? Okay, when did it come out, Sarita? I think just now, today itself it came out, is it? Because I checked today morning also, I didn't see any notification in the CSIR site. Okay, okay, all right. So nothing to worry. You have time, but if any concepts are still yet to be studied, then probably you are lucky to get time to study those. March 1st means you have complete 1 month. Okay, so that's in a way a good thing. That's nice. But the next exam probably will be in September only, then, if this is in March. September or probably in December only it can happen. I'm not sure in that case. Generally, the June exam sometimes they will conduct in September if the December exam doesn't happen on time. So let's see what happens. Okay. So you are saying option 2. Okay, Satyam is saying option 2. Anyone else wants to try this? Okay. So no worries, I will explain these to you. Option 2 is correct answer you're saying. Okay, no worries on that. See, this will be on the concept that when you have any component out of these five components, when they are added, first of all, oxygen consumption means what? Oxygen consumption means that they will be happen. That means the electron transport chain is already taking place. Okay, this is what it means when it says that the oxygen is getting consumed. Okay, so first of all, have a watch out. Oxygen getting consumed means that that the oxygen concentration is getting decreased. Okay, so be careful of what is written in the y-axis. And oxygen is getting consumed means as the electron transport chain is proceeding along with the time, now one components are added here, and we can see there is the increase of oxygen consumption. Okay, initially there is no increase, no matter how much time you keep it, but after the addition of component 1 it suddenly increases a lot. So that's why we can assume that this might be some kind of activator, which is some kind of substrate, which is providing the electrons, or probably electrons were there, because ultimately electron transport chain means electrons should be transferred, right? And the final electron acceptor after the complex 4 is oxygen, which converts in the water. Now, this activator or this substrate, complex 1, it is the substrate concentration what we have if we add it. So, here succinate is the electron source which can give the electrons to FAD. That is also a part of the electron transport chain. Or else ADP and PI. That means either of these two will be 1, because none of these are activators. Cyanide, I told you it is complex 4 inhibitor. Dinitrophenol, I told you it's an uncoupler. So both processes it will make it separate. Oligomycin, it's an ATPase inhibitor. Okay. So ATPase inhibitor means F0 F1 particle will be blocked. But here it is not asking about ATP synthesis. It is asking about oxygen consumption. Okay. So that is why option 1 can be either A or E. So we have A here, we have A here, we have A here. It can be any of these three. Okay. Next, when we add component 2, we can see that oxygen consumption has decreased little bit. Okay. Now, when can this happen? See, if we use cyanide, then it will completely stop. But if it, you see, it's not a straight line like this. It has just decreased little bit. Okay, not little bit. It has decreased a lot, but it is going on at a basal level. It is still going on. Okay. So when is this possible? When the electron chain is taking place, the H plus ions are also accommodating on the other surface, but this is not working, because of which there is a saturation of H plus and no more H plus is able to pass on the other side. That is when the entire electron transport chain will slow down a little bit, until and unless some of the H plus goes back from the outer mitochondrial membrane into the cytoplasm, or some of them yeah, so that's the only way they can go out if the F0 F1 is not working, or some molecule is binding with H plus. Only in that case the H plus ion will decrease little bit, and then the ATP synthesis can continue. Okay, otherwise there is a saturation which is built, that's why it has slowed down. So 2 has to be oligomycin, 1 has to be C. Sorry, 2 has to be C. So 2C means it is option 4. Yes, that's right, Sarita. Very good. That's the right answer. I hope you understood, Rinku and Satyam, why 2 won't be the answer. Okay. Now see, upon adding 3, this is in the same tube that we are adding component 3. After adding 3, you can see a sharp increase again in the oxygen consumption. Okay. So when there is a sharp increase again, which means that whatever saturation was reached, that H plus ion concentration, because this was not functioning, now this H plus ion gradient is removed. So who can remove this H plus ion gradient? Something which can bind with this, something which can sequester with this, so that when the H plus ions are removed, again the electron transport chain can happen at a normal pace, even if the ATP synthesis is not taking place. Okay, so before adding the uncoupler, the two processes were coupled. That is why if this was not working, the electron chain was slowing down. But after adding the uncoupler, even if this is not working, the electron transport continues to take place. Okay, so 3 has to be an uncoupler, which is dinitrophenol. So the correct answer is option 4. Clear? So these are some of the activators. Glutamate and malate, they will contribute the electrons to NADH, and NADH gives to complex 1, then to ubiquinone, then to complex 3, then to cytochrome C, and then to complex 4, finally giving it to oxygen to form water. And if succinate gives the electrons, it gives to complex 2, complex 2 to complex 3, complex 1 in that case is not touched. Again, complex 3 to cytochrome C to 4, 4 to oxygen, and finally forming water. Okay. So, yeah, if you are watching this still, Tulika, this is what is the Lakshya batch I was talking about. This is for CSIR NET 2025, whenever it is taking place, and also GATE 2026 exam. Already this February GATE will be there. Then the next exam, it happens yearly, only in the next year. This is the Lakshya batch where we will provide unlimited classes for these two courses. Okay. And with such a dedicated preparation, definitely you will clear. It is advised that you take coaching from experts on this subject, and that too who have track record of clearing, helping the students clear CSIR for years and years. You can see the Biotecnika testimonials, you will understand how many students are benefiting with our courses, right? And then there is 24/7 chat support also. If you have any doubt, we are there to clear, so that you can study seamlessly without any interruptions. We have a dedicated study portal where all the video recordings of lectures and the PPTs we provide to you so that you can study properly. Ebook library is also there. The test series and the Conceptica series, which is the revision tools, and that's very beneficial in case you're preparing for this February exam. They are like Conceptica wheel, points in a circular format, table, concept table, which are comparative tables comparing important concepts and terminologies of CSIR. Those all will be given access to, and along with that you're also getting free molecular biology internship. As you know, this kind of workshops, hands-on internships, if you are completing it along with your regular courses, these are an add-on to your resume. So Biotecnika conducts them now and then often, so you should be a part of it. But we have limited seats for that, so you should contact our toll-free number or else our office number. You can contact on working hours. Toll-free number you can contact anytime. Okay. Now let's go to the next question. Yeah. Anyone wants to answer this question? This is also a part C question only. Yeah. Anyone wants to try this? Okay. Sarita says option 3. See, already we discussed little bit about this. So anyone else? See, increased blood glucose. Okay, Satyam says option 1. Okay. See, glycolysis is the breakdown of glucose, and gluconeogenesis is the making of new molecules of glucose from non-carbohydrate sources. Okay, and glycogen metabolism also. Okay, glycogen metabolism means if too much of glucose is there, you think about if too much of food is there in your kitchen, then some of it you will consume and some of it you will use it, some of it you will store it in the kitchen, isn't it? So similarly, if glucose level is high, some of it will be broken down to convert into pyruvate and some of it will be stored as glycogen. You know that glucose polymer is glycogen. Synthesis of glycogen is called as glycogenesis, which is catalyzed by the enzyme glycogen synthase. Okay, Lakshmi also says option 1. Let's have a look. And the breaking down of glycogen will happen when glucose concentration is less. Okay, if glucose is less, then glycogen will be broken down to make the glucose molecules so that it can be utilized in the so that it can be utilized again in the cellular work or cellular metabolism. Okay, it can be broken down to provide energy. And if glucose is less, then gluconeogenesis also happens. Neo means new molecules of glucose will be synthesized. Question is from where they will be synthesized. See, if glucose is not there, then what is available is are the proteins and the lipids, isn't it? Now, there are critical organs like heart, muscles, which cannot stop functioning, and brain, they will require they will require this ATP or energy, which will be released from carbohydrate metabolism, so that they can function. But if glucose is not there, then some source at least should give glucose, right? So the protein and fatty acids in our body, lipids, they will break down to give, they will get broken down and they will get converted into glucose. So making of glucose from non-carbohydrate sources, what is gluconeogenesis, right? Now here are a few sentences regarding that rate. Increased blood glucose would decrease gluconeogenesis. Increased blood glucose means glucose is already there, right? So obviously, new molecule of glucose is not required. So that makes sense that it will decrease gluconeogenesis and it will try to store some of the glucose. Okay, so it will increase the glycogen synthesis as polymer. So option 1 is correct. That's it. You have your option 1 already there. Yeah, very good. Satyam, Lakshmi, Rinku, your answers are right. I hope Sarita you got it now. Increased level of fructose 1 phosphate, it inhibits glycolysis. Fructose 1,6 bisphosphate, it is intermediate in case of the glycolysis. Okay, so if you have increased level, then it will not inhibit it. It will further proceed with the glycolysis because formation of 1,6 bisphosphate is the committed step in glycolysis. Once this is made, it will definitely go and complete the pyruvate. So it will not inhibit it. Increased blood glucagon inhibits glycogen synthesis. When is glucagon released? Glucagon hormone is released when glucose level is less. When glucose level is more, insulin will be released. If glucose level is less, then glucagon and epinephrine will be more in concentration. So that means if glucose level is less, we don't want the glycogen to be synthesized. So this is correct, that it will inhibit the glycogen breakdown and it will stimulate the glycogen, it will inhibit the glycogen synthesis and it will stimulate the glycogen breakdown. So that also is actually correct. I don't know why they have given such kind of options. Okay, so option 3 also seem to be correct, but probably I have taken the wrong options here. Okay, maybe the options are something else, like here the answer is 1 and 3 seems to be the correct answer. Okay, so probably it is A and C like that, but by mistake, I think here some other options have come. But anyways, option 1 and option 3 is correct. Let's see if option 4 is correct. Increase in AMP levels. AMP level is more means it's an indication that glycolysis is required because energy should be produced. Okay, so it will inhibit the glycolysis. That is again wrong because AMP level is high means it is going to promote glycolysis so that energy can be synthesized from the catabolic process. Okay, and it will stimulate gluconeogenesis. No, even this is wrong. So option 1 and 3 is right because if glucagon is high, it means glucose is less. And if glucose is less, why will glycogen be synthesized? In that case, glycogen synthesis will not be there. Okay, and glycogen breakdown will be promoted so that we can get new molecules of glucose. All right, so yeah, I think I have taken some other options. So answer key is 2 means here it would have been option 1 and 3, that is statement A and C. By mistake these have appeared over here. Okay, anyways, it is option A and C which are correct. I hope the confusion is cleared. All right. All right, the next question again from metabolism. So this is regarding cyanide. Already we spoke about it, but there are some reasonings which are given for this. Again, anyone wants to try? Okay, Sarita, you are saying option 2. Okay, Satyam also says option 2. Yes. So look at the cyanide effect over here. Cyanide main effect is to inhibit the oxidative phosphorylation because it is inhibiting complex 4. We know a process where oxygen is utilized for the production of essential cellular energy sources. So since B is saying that it inhibits electron transport chain and thus oxidative phosphorylation, that's why B is correct. It does so by reversibly binding to the ferric ions of the enzyme cytochrome C oxidase. Cytochrome molecules, generally they will be using iron, which can be converted into the ferrous form into ferric form. They can undergo oxidation reduction in order to take the electrons and again pass it on to the next molecule, whatever it is. But if this process is blocked, this ferric ions, they will bind with cyanide, will bind with these ferric ions, then they don't allow the cytochrome C oxidase to function properly. So that is why the entire, even if it is the last molecule to be blocked, but the entire electron transport chain will be inhibited. So that is why also it is first option is correct, that it binds to the heme A3 in mitochondrial cytochrome C oxidase, that is in complex 4. So A and B is the right answer. Yeah, 2 is right, correct. Sarita and Satyam, your answer is right. Okay. It directly blocks replication, no. And it blocks the protein trafficking inside mitochondria by affecting TIMTOM, no. That is also not correct. Okay. So yeah, your option is right. Answer 2 is the right answer. Answer key 2 is the correct option. Yeah, that's right. Um, for research in biotechnology, KK, the best exam for research in biotechnology is by default the CSIR NET exam. Okay, that is the best and number one exam which is nationally accepted in most reputed institutes of India. If you clear this exam, not only you will get the admission after clearing interview in those institutes, you will get admission to PhD research, and not only that, you will also get fellowship per month. You will get fellowship, a huge amount of fellowship. Okay, so you can earn and pursue PhD at the same time. So definitely you should aim for the CSIR NET exam. Okay. Yeah, Rinku, very good. That's the right answer. Even Satyam also. Lakshmi also, your answer is right. Very good. Okay, let's go to enough of carbohydrate metabolism. This question is from June 2024, the recent one, the latest exam. They have given a question on fatty acid metabolism. So this is an arrangement type of question where you have to arrange the processes one after the other. Okay. So I hope you people are aware that there is a webinar on 2nd of February. Yes, are you people aware? 2nd of February, 11 to 12:30. There is a CSIR NET webinar. Do attend it without fail if you are preparing for this 2025 exam. Now it is in March. So first March exam, if you are preparing, then definitely these are the last minute tips and tricks which are going to help you in performing in the exam better. Okay, so kindly, you should attend it. Okay, do not miss it. Yes, anyone wants to try this? What are the steps that are involved in oxidation of lipids? We know that lipids are broken down for energy, and the process that is used is beta oxidation, right? Where two carbons is removed at a time as acetyl CoA. But under beta oxidation, what are the steps? Okay. Sarita and Satyam both say option 1. Okay, let's have a look. See, anyways, when we have the fatty acid, okay, first of all, the fatty acid, it has to be conjugated to acetyl CoA, isn't it? So this should be one of the first things that it should be activated, where an ATP will be required. Okay, but even before that, so we have some more steps which will follow. So you can see the sequence. The triacylglycerol will break into the fatty acid. Then, as I said, the fatty acid will be bind with the coenzyme A. Okay, so ATP will be required. Before that, the triacylglycerol will be breaking down to give the fatty acids. Then only it can bind to coenzyme A. Then only it can become fatty acyl CoA. Now that it has become fatty acyl CoA, it cannot directly cross the membrane of mitochondria. Okay, so there is something called as carnitine shuttle, which will associate with the fatty acyl, not the CoA part, only the acyl part. Okay, it will bind with the acyl fatty acid part, which is a long chain fatty acid. The carnitine will bind with it, CoA will stay outside only, and then this fatty acyl carnitine will pass through acyl carnitine translocase. It will help it to enter into the inner mitochondrial membrane. As I said, not much is inner mitochondrial membrane is more or less an impermeable membrane, so nothing much can pass through it. Okay, so once the fatty acyl carnitine is inside the membrane, another CoA molecule inside the mitochondria binds with it, and we have the fatty acyl CoA. Once again, carnitine comes out, so this is just helping in the shuttle. Okay, now once we have the fatty acyl CoA, the first step is oxidation, where the electrons will be released and it will be given to FADH2. All right, so we have after the oxidation, we have the hydration. After hydration, one more step of oxidation, and the last step will be the thiolysis, where the acetyl CoA is broken, one molecule, and another CoA will be attached to the rest of the fatty acid chain. So the end product of this before going into another cycle is another fatty acid which is shortened by two groups. Okay, so that is why the correct step is first the hydrolysis of triacylglycerol by lipase will be taking place, which is breaking the triacylglycerols to release the fatty acids. Then it will be D, because activation of fatty acid by conjugating. Okay, after that we have the reaction of the fatty acyl CoA with carnitine. So we have A. Then we have the oxidation, but it is not mentioned. So we'll go to the next step, which is hydration, that is E. And finally, it ends with thiolysis. So yes, very good everyone. Satyam, Sarita, Rinku, your answers are right. Option 1 is the correct answer. Okay. All right. This is now from nucleotide biosynthesis. So can you tell me which answer is right over here regarding nucleotide biosynthesis? Okay, Sarita says option 1. Yes. How about others? Anyone else wants to try this? See, we know purine and pyrimidine nucleotides, they can be made through two processes. One is the de novo synthesis, which is the synthesis of them from scratch, where one by one the carbons and the nitrogen in the skeleton are added. So these mentioned below are all with respect to de novo synthesis. Okay, each of the carbon and nitrogen in the molecule of nucleotides, they are contributed by the amino acids, and you should know by heart. You should know, memorize it properly. What are those carbons and nitrogen which contribute? Okay, so biosynthesis of both purine and pyrimidine nucleotides, they will begin with ribose 5-phosphate and purine rings are built on it. The main difference between the purine and pyrimidine is that initially the ribose sugar will be made. Okay, and for purine, on the ribose sugar, whatever is the ribose sugar, on top of that the purine atoms will start to get added. Okay, but for pyrimidine the difference is that individually the ring will be made, and then it will be already ribose sugar will be there, then it will be added to the pyrimidine ring. This is the main difference which is there. Okay, then the next, so it says biosynthesis of both purine and pyrimidine begin with ribose 5-phosphate and purine or pyrimidine rings are built on it. So this is not correct. Okay, we have to select which is the incorrect statement. So option 1 seems correct. Okay, because that's not the case. That's a difference. Then the first purine nucleotide biosynthesized by the de novo pathway is iosinic acid or ionosine monophosphate. Basically, it is ionosine monophosphate. And yes, that's right. For purines, this is what is produced first, and from here there is one or two steps more with which you get your purines, adenine and guanine. Okay, from inosine monophosphate only you will get those two. So this is correct statement. First pyrimidine synthesized by de novo pathway is orotidilic acid. Even this is correct, because from here some changes happen which makes it come to uridine, and from uridine you get cytosine, and then uridine undergoes some changes to make thymine. Then TMP is synthesized as deoxy TMP from deoxy UMP by thymidylate synthesis. Even this is a correct statement. Okay, because when you get deoxy UMP, see, UMP directly cannot produce UMP directly, you cannot have TMP. That is not possible. Okay, uridine monophosphate can give rise to CMP with just one change, but directly it cannot make TMP. For this, it has to first get converted into deoxy UMP. Okay, then only it can give TMP. Can any one of you here tell me which is a very common inhibitor of this process? Which is a common inhibitor of this de novo process it can block the purine synthesis also, and this pyrimidine synthesis also it can block. That is conversion of this UMP to TMP, DUMP to DTMP. Basically this thiamidylate synthetase, what is there, this enzyme can be blocked by a very common molecule or a very common chemical. Can anyone tell me what that is? And it can also block the purine synthesis as well. Anyone? No one? See, again, I think there is a little bit lack on your end for the inhibitors and activator names. So you need to work on it little bit more. Okay. Yeah, Satyam, your answer is right, but I am asking the name of one common inhibitor which blocks the de novo synthesis of both purine and pyrimidine. Yes, yes, Lakshmi. The live class will be accessible. You can see the recording will be there in Biotecnika channel. If you want to see it later, you can definitely see. Okay. Okay. So, have you guys heard of this term methotrexate? Heard or not? Yes. Methotrexate or aminopterin. Have you heard of this aminopterin? Yes, so these are the blockers. Okay, these are the popular blockers. Actually, both of these processes, that is purine and pyrimidine, they require this enzyme dihydrofolate reductase DHFR. Okay, for the transfer of either the methyl group or formyl group in purine and methyl group in pyrimidine. So these aminopterin blocks this enzyme, because of which the de novo process is blocked. That's the entire concept of hat media, isn't it? So in that case, yeah, yeah, correct. So that is how you get the hat media. And hat media, the if the de novo pathway is blocked, then at least the purines and pyrimidine should be synthesized by the salvage pathway, isn't it? So salvage pathway is an alternative pathway of purine pyrimidine synthesis, which recycles the existing nucleotides. Okay, so some things we will ingest it earlier. So that's why the salvage pathway, for it to continue, it needs some precursor molecules, that is hypoxanthine and thiamidine, which will be able to give rise to the purines and pyrimidines in case the synthesis is not happening through the de novo pathway. So hat media is used for selection. Any fused cells, it is used for the selection of the fused cells in case those cells are not able to grow through de novo. Okay, remember that it is useful for monoclonal antibodies. Already I have told you the process. This is the pathway for pyrimidine synthesis and pathway for purine synthesis. Okay. So I was telling you, no, see this thiamidylate synthetase, it is blocked by fluorouracil also. And we have this thiamidine, means the methyl group. See, UMP and TMP, what is the difference between them? Thymine is methylated uracil. So a methyl group has to be transferred. And that methyl group is transferred by something called as methylene THF. So THF brings the methylene group. Okay, methyl group. If THF is blocked, and THF we get from DHF, which is dihydrofolate, from there we get tetrahydrofolate by the enzyme dihydrofolate reductase. If this is blocked, then this THF is blocked. In that case, this enzyme is blocked. We will not get the pyrimidine from de novo. Okay, this is also a common topic from which questions may come. Yeah, this is your last question from metabolism. Then we'll look at one or two protein based questions. Kindly try to answer this. So biosynthesis of proteins and nucleic acids from precursors result in the production of chemical energy in the form of ATP, NADH, NADPH, and FADH2. Then something regarding thermodynamics also, spontaneity of a reaction in cells, it does not depend whether the delta G0 is positive or negative. Any guesses? Both oxidative phosphorylation. Yeah, very good. Satyam, that is the right answer. Did you know it from before or you answered just now? Because this statement, it is usually confusing for students. Okay, this statement is usually confusing because people will relate spontaneity with delta G. But the small trick that they have used over here is it is not delta G, it is delta G0. Delta G0 means it is standard free energy change. Okay, whereas delta G, it is only the regular free energy change. Okay, so standard free energy change means in standard conditions, 1 molar concentration of substrate and products, then temperature also, uh, pressure also, everything is fixed at standard conditions. Now, if anything is happening at standard condition, then how can it predict spontaneity? Spontaneity is that in regular cellular conditions, what will be the, like in what direction this the reaction will take place. Spontaneity means whether the reaction can happen on its own. Now that will be decided only by the by how much in regular conditions the reaction is proceeding and not at standard state. So that's why this is only the incorrect option. Yes, very good, Satyam. The spontaneity of a reaction in cells depends on the change in Gibbs free energy, where a negative delta G indicates a spontaneous reaction and a positive indicates a non spontaneous. Very good, Satyam. Correct. Okay. So that's why B is wrong. Now, the other things, yes, if you synthesize proteins, nucleic acids from precursor, that is nothing but anabolic reactions, and anabolic reactions, they will result in the production of chemical energy in the form of ATP, NADH, NADPH, and the FADH2, all that. Okay. No, 1 minute. Sorry, B is incorrect statement. You're supposed to mark all the incorrect statement. No, sorry, option 4 may not be the answer, because even A is wrong. You have to mark all the A statements. So let's see out of these two which is correct. Now, only chemical potential energy contributes to proton motive force in mitochondria. Yeah, this is not correct. No, wait, let me just read this. Both oxidative phosphorylation and photophosphorylation involve oxidation of water to oxygen. Okay, C is not correct. Okay, because in oxidative phosphorylation, we have the electrons released from the bigger molecules, that is the macromolecules, and ultimately through the electron transport chain, the electrons are given to oxygen, which converts into water. Okay, so that's what happens in oxidative phosphorylation. It is the since electrons are received, so gain of electrons means it is reduction, which means oxygen is reduced to water in case of oxidative phosphorylation. But in case of photophosphorylation, which is photosynthesis, the water will be broken down to oxygen plus protons, okay, protons and electrons, and those are given to the electron transport, not electron transport chain, the photosystem, PS1 and PS2, it will be given to that. Okay, so that is how they will start the process of the photosystem and electron transfer. So electron transfer is common in both of them, but here, yes, water loses the electron, so it is oxidized to oxygen. This is correct in photophosphorylation, but not in oxidative phosphorylation. So, yeah, A, B, and C are all incorrect statements. Okay, only D is the correct statement. So, option 1 is the right answer. Clear? You have to mark all the incorrect statement. So, A is also wrong. Okay, I'm really very sorry. This is does not depend. It says does not depend. Okay, yeah. Sarita, your answer is only right. A, C, and D is only correct answer. You said it correctly. Satyam also. So incorrect because it does not depend means this is a correct statement. Okay, I thought it is written depends. So this is a correct statement. Rest all is wrong. This is wrong, this is wrong, and even this is also wrong, because only chemical potential energy it contributes to proton motive force. No. What is the other thing which contributes to the proton motive force? It is also the chemiosmosis. That means osmosis power also contributes to the proton motive force, because of which this entire oxidative phosphorylation, which is the ATP synthesis along with electron transport chain, it is called as chemiosmosis. Okay, so chemiosmosis, both of them will be promoting the electron transport chain to build up the proton gradient, because of which it will be building up the proton gradient force. Okay, so chemical potential energy and osmosis will contribute to proton motive force. But since it is written only, that is why this is wrong. So C is wrong, A is wrong, D is wrong. Option 4 is the right answer. Yes, correct. Okay. So these are the differences between the oxidative phosphorylation and photophosphorylation. All right, let's discuss one question on the proteolysis. Um, we have been given two amino acid sequences. This is one sequence and this is another sequence. Basically, these are two polypeptides. Okay, and some experiments were performed on the peptide X. So, on the peptide sample X, in experiment 1, treatment of this X with DTT, followed by DTT is a reducing agent which will break the disulfide bonds, followed by blocking of the free sulfhydryl groups. So, once the sulfhydryl bonds are broken, these groups cannot come back together, so those will be blocked. In that case, it yielded two polypeptides whose sequences I have given like this. Okay, but in the second experiment, the protein, same protein X, instead of being treated with DTT, it is now treated with chymotrypsin, which is a protease that will cleave the carboxy terminal of aromatic residues. Okay, so the amino acid composition of 5 peptides obtained from this experiment are given. If from experiment 2 you got all these different fragments. Okay, now how can we conclude how are the cysteines linked? Okay, so the cysteines are marked here, A3, A6 and A9 in one of the polypeptide chain, and in the second B polypeptide chain it is the B2 and B8. So they are asking how are these cysteines linked together, right? Okay, Satyam is saying option 3. So let's have a look how to solve this question. Okay, see, just a minute. Yeah. So you look at the diagram over here. Okay, look at the diagram. I told you DTT, this is the case where it's a result of the first experiment. So DTT treatment cleaves the disulfide bonds in a protein. And since post DTT treatment, the free sulfhydryl groups were blocked, so they could not join back again. Okay, so that is why they give these two subunits, that is A and B of the protein X. So clearly, protein X is a dimer. Okay, and in the second experiment, this intact protein without any DTT treatment, that means the entire polypeptide chain, that means including both the polypeptide chains, the entire protein, it was cleaved with chymotrypsin, only with chymotrypsin. And we know chymotrypsin, it breaks the peptide bonds near the aromatic residues, which is phenylalanine, tryptophan, and tyrosine. Okay. So this should yield 7 fragments as given in the figure. If you treat with, means where all we will be having the cleavages, if you treat with chymotrypsin, so before this you can see this is the red molecule, red lines you can see, no? So these red lines are actually the places of the C terminal of aromatic residue, that is either phenylalanine, tyrosine or tryptophan. So here one cleavage will be there. Okay, see this is an intact protein. Now imagine this is an intact protein. Okay, so if you treat with chymotrypsin, then where all the cleavage will take place, so that you get these fragments. So one will be cleaved here. Okay, one will be cleaved here, wherever the aromatic residues are there, they will be cleaved. One will be cleaved here. Okay, okay, and one will be cleaved here beside the tryptophan, beside the phenylalanine. So totally you are getting 1 2 3 4 5 6 7. Okay, but we are getting only 5 fragments. Ideally, the intact protein, we should have got 7 fragments if you would have treated with DTT. If you would have treated with chymotrypsin. Intact protein. That means including its disulfide links. But we got only 5 fragments. Which means what? So first composition, you can see alanine and phenylalanine. This is okay. It is the component A. So this is fine. Okay. Second composition is a mixture of both fragment B and D. So you can see B component and D component also. Okay. Because you can see cysteine is present here also. So two cysteines you're getting. Methionine, one methionine is there. Tyrosine, one tyrosine is there, and one aspargine is there. So it's a combination of B and D, and in B and D you have cysteines also, which means A3 and A9 should be connected together. Okay. Then the third composition, that is cysteine, glycine and lysine. So this is completely of fragment G. Already you have a separate one. So since you have got a separate fragment like this, we can conclude that the B8 of cysteine hasn't been connected with any disulfide linkage. Okay, so these are like individual fragments. Okay. Fourth composition, it is a mixture. You can see many amino acids, no? So if you look closely, it is a mixture of both C and E. So that is cysteine is present here, cysteine is present here also. So two cysteine, one leucine, two tryptophan, and one valine. Okay, so that means that the cysteine in this C fragment, which is the A6, should be bonded with the cysteine in the E fragment, which is cysteine B2. Okay, so they should be bonded together. Again, if you look at the fifth component, isoleucine, phenylalanine and valine, it is nothing but the F fragment. Individually, it is the F fragment. So G fragment is the only one which has a cysteine residue, but it is not disulfide linked. It is independent. Okay, so that's why we can say the second answer is right. A3, A9, and B2 and A6. Is it clear? Satyam and Rinku, others also, did you understand how to solve this kind of questions? Okay. So that's it from my end. Thank you. Next, I'll come for your Brahmastra series. On next week, I'll come. Next week, we will be discussing Unit 5, Developmental Biology. That's all discussion of Unit 1 as of now. Okay. Next class, it will be Developmental Biology Unit 5. So you can revise so that we can discuss the questions. Thank you, everyone. Good night.